Hi again, everybody. Today we're gonna continue on into the fat soluble vitamin section. So, fat soluble vitamins have it's basically just four vitamins, so much smaller than what we saw for the water solubles. So, the way that I usually remember fat solubles is ADEC. It's A, D, E, and K. So, yeah, we'll just be doing those four, and then we'll have another unit like midterm after that. So, first, starting with vitamin A, um, obviously, usually when I teach this in person, I like to have like a little bit of a discussion about what people kind of already know about vitamin A and the carotenoids. So, if you just kind of think quickly to yourself, what foods provide carotenoids? Usually, most people know it's like carrots, that type of thing, orange, red vegetables, fruits, that type of thing. And then, what does vitamin A do in the body? So, why do we need it? Typically, people know that it's related to eye health or night blindness, so that's one of the major things that are mentioned. And then some people know it's related to like growth and cell proliferation and stuff like that. So, yeah, those are kind of the main things that people tend to know about vitamin A going into the course. So, structure of vitamin A. Basically, vitamin A has a lot of different vitamins, which we're going to go through. And it's a bunch of compounds that possess the biological activity of all trans retinol, OL retinol. There's also retinal, so I'll try to make that distinction because I know it's kind of hard to hear the distinction. And basically, all these compounds kind of look the same. They have this ring structure here, so a beta adenine ring, and then that's attached to a polyunsaturated side chain. So they pretty much look like this ring and then this like chain. And then what differentiates the different vitamins of vitamin A is that they have this little additional group here, basically stuck onto the end of that polyunsaturated side chain. So in the case of the first vitamin, or retinoid as it's also called, is retinol. That one has an alcohol group attached here, so an alcohol group. The second kind is retinal, Al. That one's different because it has an aldehyde group attached. So I wrote that right there. Then there's also retinoic acid. That one has a carboxyl group at the end. And then the fourth retinoid is something called retinol ester. And retinol ester has a palmitate, which, as you know, is a fat. You probably remember this from Dr. Song's class, but it's a fat, so basically it has a fat attached. And then also just to remind you from like your biochem classes, an ester just means that there's a fat attached to it. It took me a really long time. I think it wasn't even until like after my undergrad, which I realized like ester or esterified just means that there's like a fat attached to it. So yeah, retinol ester means a fat attached, and specifically it's palmitate that's attached right there. So as we go through this, I'm also going to be discussing the carotenoids. So as we go through structure, I'll do vitamin A first, and the carotenoids, and then same with function and DRIs, just to kind of so we can do like a cross comparison between them. So structure of carotenoids, it's interesting. Well, I'll show you in a second. But the main thing I want you to remember is that carotenoids are precursors to vitamin A. So you might have heard them referred to as provitamin A before. Provitamin A is another way to say a precursor. And then characteristics of the carotenoids, they're fat soluble, so lipid soluble. They generally have a red, yellow, or orange pigment. And then there's a whole bunch of them in nature. So there's been about 600 identified so far, but only about 60 can act as like provitamin A or can be basically converted into retinol. So, and then we're actually just going to focus on about two or three of them in this lecture. So if you look at the structure of carotenoids, so this is beta carotene down here, you'll see it has two rings in this chain. What does it kind of look like to you from what we just looked at? Basically, beta carotene is simply two retinals attached. So if you were to snap beta carotene in two, you'd basically get two vitamin A's, specifically two retinals. So there's retinal, what we saw before, and then here's beta carotene. You literally cut it in two and you'd get two of these guys. So just to kind of go through what we discussed so far, because I know we've talked about a bunch of different compounds. So vitamin A, or preformed vitamin A, is also referred to as the retinoids, which is a group of compounds such as retinol, retinal, retinoic acid, and retinol ester. So it's those four that we talked about. That's when I showed you like how the different attachments basically on the end. And then precursor vitamin A's are the carotenoids. And then the main one is beta carotene. That one is basically the main one because it is most likely to become vitamin A. Like it's most easily, I guess, kind of snapped into and made into vitamin A. But then there's other ones that have vitamin A capacity as well. Lycopene and zeaxanthin basically kind of perform the functions of the carotenoids, and they're considered precursor vitamin A's as well. So food sources first now are back to vitamin A, because the food sources for vitamin A and carotenoids are very different. For vitamin A, it's pretty much animal products. So if you look here, it's like meat, milk, cheese, like dairy and meats pretty much have very high sources of vitamin A. If you want to write down the DRIs, it's an RDA. It's in micrograms, and it's 700 for women and 900 micrograms per day for men. So the kind of crazy thing here is if you like liver contains like 10 times or more than 10 times what we need just in one serving. So this is actually kind of above the TUL even. It's kind of at a toxic level. If it's for people that, if you consume liver occasionally, it's not that big of a deal, but if you're eating it once a week, it can actually be kind of toxify you in different ways, which we'll discuss throughout the class. But particularly with vitamin A, you're getting super high doses of vitamin A because a lot of it is stored in the liver. Other places we get vitamin A are like fortified and enriched products. So from what I can tell, again, using like Cheerios is just like one point of comparison. It's usually about 20 to 30% of your vitamin A needs for the day are in fortified grain products. So if you see anything higher, let me know. But that's in generally just looking at food labels. That's kind of what I've seen. And then RAEs, which I have up here. So that's called a retinol activity equivalent. Basically, that is equivalent to just 1 microgram of retinol. And I'll show you, this will become a bit more clear in the DRI slides. This is a way to kind of show you how much retinol can be made from beta carotene. And then also how it relates to the international units. You've probably seen like on fat soluble vitamins, they report them as IUs, international units. So again, we'll talk about that mainly in the DRI slides, but just to point out right away, those international units are only used for fat soluble vitamins. So remember, we didn't mention those at all in the water soluble portion, water soluble vitamins. So yeah, I use just for fat soluble vitamins. You won't see them for minerals or water soluble or anything else. So when we consume retinoids, so basically vitamin A, those different types of vitamin A, you take it in, or it's in food attached to fat. So specifically, it's attached to a palmitate in food. And so we've already kind of talked about this is a specific retinol ester, so retinol palmitate. That's like the fourth one we went through. And this is kind of like the storage form of vitamin A. So it's a way that it's stored in the body, and then it's also the way it's stored in animals' bodies, basically, like in the storage form that we'd get from like meat and stuff like that. So just know that in foods, vitamin A is stored bound to a fat. And what does cooking do to vitamin A and carotenoids? In general, they're pretty susceptible to oxidation and heat. So vitamin A and carotenoids will undergo oxidation if exposed to heat. So you can basically destroy it. But it usually happens at pretty high temperatures, so above 200 degrees Celsius. So kind of the same thing that we saw with like water solubles, where you're really losing it at really high temperatures, at like pressure cooking, grilling, frying, that type of thing. And then another kind of interesting thing that happens with the fat soluble vitamins is that if you were to fry something in fat, because they are fat soluble, so if you're like deep frying something, you're actually going to lose a lot of that vitamin into the fat. If you remember with water soluble vitamins, if you're boiling vegetables, the water soluble vitamins will like seep into the water. Well, the fat soluble vitamins will actually seep into the fat. So I probably wouldn't recommend recouping like you don't want to like add all that extra oil to your meal or whatever to recoup the vitamins. So I definitely recommend not doing that. But just so you know, yeah, it actually, since it is lipid soluble, it will actually just kind of leak into the fat. Exposure to air and light is another thing that can kind of destroy vitamin A. So it will undergo oxidation if exposed to air and light, and it's about a 10 to 20% loss after 48 hours. And this is particularly important with regards to milk. So remember, milk has vitamin A in it. And that's why we usually see milk being stored in kind of opaque or translucent containers. It's really to protect the vitamin A from light oxidation. Interestingly, kind of the more expensive milks, like the Oberweis and the more of the like organic brands and stuff like that, for some reason they don't store them in, it's all like clear containers. And I have noticed that the expiration date on those is a little bit sooner than the other types of milks. I'm not sure if it's because of the container. I don't really know why they chose not to put it in an opaque container, but yeah, that usually comes up when I'm teaching this class in person. So I don't know, haven't been able to figure out why they do that. So, food sources of the carotenoids in particular. So, now moving on to that totally different kind of group of compounds again. Basically, for that one, it's all plant products. So, you're going to find it in a whole variety of like fruits and vegetables, pretty much, particularly in ones that are bright yellow and orange. And then carotenoids are also found in green vegetables. However, the chlorophyll is a really strong pigment, so it actually kind of masks that orange color. So the orange color's there, but if you have something with a ton of like green chlorophyll in it, it will just mask it. So yeah, in general, from fruits and vegetables, there are no DRIs for carotenoids, so I don't have anything to give you here to do any comparisons. Digestion of now looking at retinoids and carotenoids together. In general, you take in carotenoids bound to proteins, and then if you remember, the retinoids are bound to fat. So what you want to do is cut the carotenoids and the retinoids off of those fats and proteins before you take them in. And that's just done by like peptidases, pepsin, other proteases, and lipases. So basically, what you end up with is free carotenoids and free retinol. So this is pretty important. Remember, in foods, the way that you consume vitamin A is as retinol palmitate. The second you cut off that palmitate, you end up with this form of vitamin A, retinol, free retinol. So this is kind of straight out of the textbook, and I know it looks confusing, but I do have a summary slide coming up in a couple slides to kind of just whittle this down a bit, because I know it looks like a lot. So carotenoids and free retinoids, remember that's what we ended up with. So they're both attached to something in food, but we cut it off the proteins and the fat, and now we have carotenoids and free retinol. The next step is that they're incorporated into micelles. So again, this is a totally different absorption pathway than what we saw with the water-soluble vitamins. Water-soluble, remember it was like passive diffusion or some energy-dependent process. When we're talking about the fat-soluble vitamins, they basically follow the same absorption pathway as fats. So micelles and chylomicrons, if you remember from Dr. Song's class. So they basically become a micelle. And remember, micelles aren't an actual like transporter. It's just this like spontaneous formation that occurs when you have things that have hydrophilic heads and hydrophobic tails. So it's just something that happens. They literally just end up forming this because the things that like water go to the outside, and then the tails that don't like water kind of stick on the inside. So they just spontaneously form these like balls basically. And then that whole component, the full micelle, is basically taken into the enterocyte. Absorption efficiency of retinol is about 70 to 90%. So we can actually absorb most of the vitamin A that we take in. However, absorption is generally, if you're consuming a very low fat diet, so less than 10 grams of fat, you can actually kind of greatly, your absorption will go down. But if you're consuming, most people do have about 10 grams of fat with a meal. That's pretty typical to have over 10 grams of fat, so that helps absorption. But this plays into the fact that people on very low fat diets, so people like really heavily restricting fat, will often end up with fat soluble vitamin deficiencies, just because you need at least 10 grams of fat to basically invoke like proper micelle formation as well. So kind of just slows down or impedes absorption with really low fat diets. Carotenoids, on the other hand, aren't that well absorbed. From fruits and vegetables, you're getting less than 5% absorbed, and then you absorb about 60% from like beta carotene supplements. So we're going to go over 4 different pathways. So beta carotene is absorbed by 2 different pathways. I'm going to show you that, 1A and 1B. Retinol has its own pathway, and then the other carotenoids, so everything else other than beta carotene, like lutein, zeaxanthin, that type of thing, they have their own pathway as well. So first, to kind of show you the first beta carotene pathway, so 1A. So what happens, remember, we broke the beta carotene off the protein, we brought it in via the micelle, so basically we're right here now. What happens then is beta carotene is then broken into 2 retinals, because remember, you can snap it in 2, makes 2 retinals. Then each retinal is converted to retinoic acid, so another vitamer, another retinoid. And then that retinoic acid will travel to the liver attached to albumin. So that's one way, beta carotene to retinal to retinoic acid. And then, just if you want to see again, really cut these things in 2 and you get those 2 retinals. Alternatively, the other pathway that beta carotene can follow is that after it's snapped into the 2 retinals, it can be converted to retinol. And it does this while it's attached to CRBP2, which is cellular retinol binding protein 2. So the retinal becomes retinol, and then it's converted to retinol palmitate, and then boards the chylomicron, and then exits the enterocyte. So retinal, you either look at the first pathway, which is it becomes retinoic acid and it's out, or alternatively, the retinal becomes retinol, then retinol palmitate, and then leaves via the chylomicron. In terms of retinol, because remember, when we get it from animal sources, we're getting basically retinol. So that's after we broke it off of the retinol palmitate. Remember, it becomes retinol. We absorb it via the micelle. Then the retinol, so basically we're here now. The retinol then binds on to the CRBP2. It then becomes retinol palmitate. Once it's retinol palmitate, it kind of boards onto the chylomicron, and then it leaves the cell in that form. So that's kind of what happens to retinol. And then the other thing that we can consume is other carotenoids. So that's like anything else other than beta carotene. So lutein, zeaxanthin, that type of thing. We'll be briefly talking about what those do in a second. Those are pretty simple. They don't undergo any metabolism. They basically just board onto the chylomicron and enter the cell. So there's no like conversions or anything for those. If you ingest lutein or zeaxanthin, they just stay like that. They board onto the chylomicron and then leave. So after leaving the enterocyte, the chylomicrons enter the lymphatic system and then the general circulation. And their purpose is really to deliver the retinol palmitate and the carotenoids to tissues. So they deliver it pretty much all over the body, Yeah, so retinol palmitate and the carotenoids were like the two major things we ended up with. And then after it's kind of floated around the body, distributing these things to different tissues, small amounts of it, so the chylomicron remnant, which still contains a little bit of vitamin A, will return to the liver for storage and metabolism of vitamin A. So these are these summary slides I was kind of mentioning that will help clarify those absorption slides. If you study nothing else in this, I would study this slide and the next slide because it kind of summarizes most of what happens with vitamin A, and it kind of just helps to clarify all the intraconversions because I know it's a little confusing. So this is the metabolism and how all these things are related. So remember, beta carotene, snap those in two, makes two retinals. That one we know. That's irreversible. So the body can't reattach these two to make beta carotene. So basically, once it's snapped in two, they stay as two retinals. Oh, sorry. Then the next thing, the retinals can become, as we saw in those absorption slides, remember the first pathway was that the retinal became retinoic acid. Once that conversion occurs, that's irreversible. The other fate of those retinals can be that it becomes retinol. So this is a reversible reaction, so the body could easily kind of transform these two things back and forth. And then retinol can then become retinol palmitate. So just remember, beta carotene becomes two retinals, retinal can become retinoic acid, or retinol can become retinol and then into retinol palmitate. And these are the reversible ones. And the reason that the body is kind of constantly forming all these different types of retinoids is because each retinoid does its own completely different function from the other ones. So retinal, this is the one that is involved in night vision. So whenever you hear vitamin A being important for eye health or vision, we're talking about retinal in particular. Retinol, its function is in growth. Retinol palmitate, as you probably guessed already, is the storage and transport form. So this was the form that we found in food. And this was also the one, if we kind of go back to all these slides, this is the one that was like floating around the body. So look, everything was made into retinol palmitate before it got on the chylomicron to be transported around the body. So yeah, really this one is the transport and storage form. And then retinoic acid, that is the one that is involved in gene expression. So I'll go over those functions in more detail now. So vitamin A, vision, gene expression, growth, those are the main things that those different retinoids do. And then carotenoids, as you probably already know, has an antioxidant function. Particularly, it's used in supplements for like eye health and heart disease. And I'll kind of go over that function and whether or not it works as well. So first, let's start with retinal. This is the one that is needed for night vision. So remember from your physiology classes that the retina is basically that inner lining on the back of the eye. So it's kind of this like blanket on the back of your eyeball on the inside that absorbs light. And the retina contains light receptors. So there's rods and cones, as you probably remember. The cones are the ones that function in the day, whereas the rods are the ones that are involved in night function. So they function during the night. They help you see in the dark. And vitamin A, the whole reason that it plays an important role in vision is that you're actually using vitamin A itself to make parts of the retina. So in particular, you're making this thing called rhodopsin. And rhodopsin is a protein found in the rods of the retina. So basically, you need vitamin A, like the actual vitamin A molecule, to see. And I'll show you exactly how that. I think a lot of you have probably learned this in physiology already, so it's more of a review. But I'll show you exactly what I mean. So the protein pigment that we're making is called rhodopsin. Rhodopsin has a cis retinal, so retinal in the cis form, attached to an opsin protein. So really, this thing stuck kind of on the retina is rhodopsin, and it's a retinal, cis retinal attached to an opsin protein. In the dark, when a flash of light hits the retina, Rhodopsin is basically that thing that we just made is cleaved, so it's broken in two. So this slide kind of summarizes it a little bit better. So here we have rhodopsin. Remember, that is found in one of the rod cells. Rhodopsin is made out of the protein opsin and then cis retinal. What happens when a flash of light hits? These two things are broken apart. So opsin is cut off, kind of leaves, and then the cis retinal is instantly made into trans retinal. So that's basically the two things that happen. Opsin is cut off, cis retinal becomes trans retinal. So in order to regain vision in the dark, you basically have to build back up this rhodopsin molecule or protein. So in order to regain vision, you simply have to reattach the opsin protein back on to retinal, but retinal has to be back in its cis form. So you need to turn that transform back into cis and then attach them. So yeah, to regain basically successful night vision, or if you can see well in the dark, it means that your body has enough vitamin A to constantly supply this, and that your body can very efficiently put these two things back together and put retinal back into its cis form. And then, yeah, that's how you know you actually have, like, that's one way that they can measure appropriate vitamin A status. So if you're, in the dark and all of a sudden, or how would I say this? If you are in a bright place and then all of a sudden you walk into a dark room and it takes you like a very long time to kind of be able to see even a little bit, that's one way that you can tell maybe that you have kind of lower vitamin A status. Whereas if your night vision kicks in, within seconds to a couple minutes, you're probably fine. But clinically, I believe that is one way that they measure it. So yeah, night blindness is basically just inadequate vitamin A intake, which leads to slow recovery in the dark. So moving on to the next retinoid, retinoic acid. Remember, that's the one that's involved in gene expression. So specifically, retinoic acid in the cis or trans form, in order to be involved in gene expression, is first transported to the nucleus bound to CRABP, and that just stands for cellular retinoic acid binding protein. Then the retinoic acid binds to nuclear receptors, so that's these guys, RAR and RXR. Retinoic acid receptors, basically, that's what it stands for. Then the complex binds to a DNA nucleotide sequence, also called RARE, which stands for retinoic acid response element, and that's found in the promoter region of genes. And then basically, that just affects mRNA transcription and protein synthesis. So the precise mechanism is still not understood, but that in general is basically how vitamin A can affect gene expression. So moving on to growth, remember the one involved in growth is retinol, OL. And specifically, they noticed that it's really important for the growth of epithelial cells. So in order to have adequate growth of epithelial cells, we need adequate amounts of vitamin A. The exact mechanism isn't clear, but they do know that cell growth is stimulated by growth factors binding to receptors. Obviously, we've learned that, I'm sure, several different times in several different classes. So they don't know the whole mechanism involved, but they do know that retinol derivatives, when a retinol binds to a cell, that increases the amount of receptors that kind of float to the surface. So then more growth factors combined. So somehow the binding of retinol to the cell, retinol, is helping to increase the amount of receptors, so more growth factor combined, so then more growth can occur, basically. And to move on to carotenoids, so what do carotenoids do? You probably knew before taking this class that they do have an antioxidant role, but I just want to say kind of flat out that it's a different role than what vitamin C does. So carotenoids don't donate electrons to basically prevent oxidation. They do something called quenching free radicals. So to back it up a little bit, carotenoids are present on the interior of membranes. So, because of what they look like, if you remember, they have like two ring structures, and then basically a long kind of chain between the two rings. So, they fit really nicely within a cell membrane. And they kind of just like, once you consume them, they're either broken into retinal, then they just do the other pathway. But if they're not, then in the case of other carotenoids, they kind of just end up in cell membranes to help perform this antioxidant function. And they possess an extended system of conjugated double bonds that makes them capable of quenching free radicals. So you may have learned about this in like biochem or something, but just to remind you, quenching is a process by which electronically excited molecules are inactivated. So where you may have seen this before is like singlet molecular oxygen. So that's when oxygen has this like higher energy, it possesses higher energy, and it's basically more reactive than ground state oxygen, which exists in that triplet form. So basically, it has a ton of excitation energy, and that energy can go on to damage like lipids and DNA. It basically like really quickly can react with other things and just damage lipids and DNA. So what you want to do, or what the body wants to do, is basically calm that thing down, I guess you could say. So that's it. Like quenching is literally like calming down oxygen. So specifically, what happens is you have this kind of excited molecule of oxygen, singlet molecular oxygen, and then it interacts with the carotenoid. The carotenoid, basically, that excitation energy is transferred to the carotenoid, and then that helps the oxygen return into its ground state. And then that carotenoid that received that energy just dissipates that energy in the form of heat. So the carotenoid itself doesn't become like excited and goes on to damage things. No, basically, it is kind of a benign thing that can like just take that excitation energy and just transfer it in the form of heat to get rid of it. So yeah, basically you're left with just kind of like normal ground state oxygen and then a carotenoid that dissipated some heat. And that's how it kind of gets rid of that potentially damaging molecule. So the main thing that you'll see carotenoids kind of marketed for is one is eye health and the other one is heart disease. So in terms of eye health, it's specifically marketed for age-related macular degeneration, and that's a major cause of blindness in the elderly. So if you don't know exactly what that is, this is kind of a nice illustration to show you what happens. So the macula is in the center of the retina. So remember, the retina is that blanket on the back of the eye. The macula is the center bit of the retina. And the macula helps to maintain central vision, but it can be basically damaged by UV rays. That's why, they're telling people to wear sunglasses and stuff if you're living in really sunny areas or in the summer, that type of thing, because UV can damage it. And then this is basically what happens with severe damage. So with normal vision, you can obviously just see straight forward. But if you have severe macular degeneration, anything you're looking directly at, you can't see. So it must be so stressful, but you basically can't see you can see your peripheral, but anything you focus directly on is blurry. So that's what macular degeneration is. And they have found that carotenoids can be used both to kind of prevent and treat it. And this is something that I'd say I would, if you have elderly clients, I think they should definitely give it a try. There's enough science out there to show that it works. So first, in terms of prevention, they have found that people that take in like higher amounts of fruits and vegetables, which would lead to higher amounts of like carotenoids, like lutein and zeaxanthin are particularly the ones that help with eye health. Those people tend to have lower risk of developing macular degeneration. So people that kind of have healthy diets all their lives, I guess not surprisingly, have lower risks of certain diseases. So in particular, macular degeneration with higher intakes of fruits and vegetables. It can also be used to treat macular degeneration. So if you take in the treatment regimen that I could find was 10 milligrams per day, and you'll see the bottles are basically like designed for eye health. So, it's not, you don't have to like buy a bunch of bottles and combine things. You can just buy the carotenoids. There's like a mix of lutein and zeaxanthin. So you want to take 10 milligrams per day for 6 months. And that has actually been able to kind of like shrink this. So it doesn't totally get rid of it, but it may turn into kind of like a smaller spot, or basically you can like people can see a little bit better. It's not so obstructive. And yeah, so generally it will take about 6 months to see this. I don't think it will completely get rid of it, but it has shown to improve macular degeneration. So yeah, in elderly patients, you could definitely give it a try. In terms of heart disease prevention, we kind of went over all of these steps before, but so this is just kind of going through those steps of atherosclerosis that I talked about, where it starts with oxidized LDL kind of sticking on, and then macrophages coming along and forming foam cells, and eventually plaques. So, what carotenoids may do is they may basically stop atherosclerosis by preventing that first step, where it may just prevent the oxidation of LDL. So again, through like quenching and that type of thing. And then that can eventually, reduce the development of atherosclerosis. It's This area is kind of contentious. There's definitely several studies that show that if you combine beta carotene with vitamin C and E, that it can kind of lower the risk of heart attacks. But then there's other ones that show that it doesn't. So I wouldn't, there's not too much harm in trying this, but you would have to do it over the course of a pretty long time to see, because like heart attacks don't just happen out of nowhere. They usually take years to develop. So if you do have clients that are interested in this, I think it's okay, but definitely work with their doctors. I'm sure they'll be on a whole host of like statins and other things as well. So work with their doctors to see if it's worth trying this route of using carotenoids to lower their atherosclerosis risk. So moving on to excretion of vitamin A, most of it is actually still excreted in the urine. So if you remember with the water-soluble vitamins, pretty much like almost like 100% of the time, or most of the time, it was excreted either as metabolites or intact in the urine. In this case, vitamin A and carotenoids are metabolized. Basically, the ring structure is opened before it's excreted, and that kind of tells the body that that vitamin A or carotenoid is like old enough for whatever it is that the body can get rid of it. And then about 40% is excreted in the feces. So that's the major thing with fat soluble vitamins. They end up kind of traveling to the liver and getting into the bile and then being excreted in that way. So vitamin A travels or may already be in the liver. It's then made into a metabolite, glucurononic acid, and then it's just excreted through the bile and then into the feces. DRIs for vitamin A, I already mentioned. So for women, it's 700 micrograms per day. So pretty small amounts, micrograms, not milligrams. And for men, it's 900. And then just to kind of show you with actual numbers now what these retinol activity equivalents mean. So 1 RAE is 1 microgram of retinol, which is 12 micrograms of beta carotene. So this basically means that in order to get 1 functional microgram of retinol, you would need to ingest or take in 12 micrograms of beta carotene. So you need a lot of beta carotene to basically start doing like the function of retinol. And then in terms of international units, because you'll see that on bottles a lot still, 1 microgram of retinol is 3.33 international units. And the weirdest thing is that these international units, I should look up actually at this stage. I haven't been able to find like a clear answer for where these came from or why they did this, but international unit conversions are different for every fat soluble vitamin. So remember how I told you only fat soluble vitamins have IUs? So if you start looking at bottles, IUs only happen on like A, D, E, and K. And then these conversion factors are different for each one. So it's like, yeah, it's definitely, it's very weird. I don't understand why it exists or what the point is. The DRIs don't mention them though. The DRIs just refer to everything as micrograms for vitamin A. So RAE, to put it more simply, just basically represents the vitamin A activity of retinol and basically how much beta carotene you would need to ingest to get that vitamin A activity. And then obviously need a lot more beta carotene because absorption is so low. So, also to point out, there are no DRIs or RDAs, any of that for carotenoids. And that's because they're not considered essential nutrients. People definitely supplement with them, for eye health, cardiovascular disease, but they're not considered essential because we can get, yeah, they basically just turn into vitamin A, and we can already get vitamin A from our diets. In terms of deficiency, this is actually, it's not super prevalent in the US, but it is very prevalent in certain countries and different countries in Africa and then also in India. And unfortunately, it mainly affects little kids, so kids under 5. And it's associated when they have a vitamin A deficiency, it's greatly associated with increased childhood mortality. So the major sign of deficiency, as you probably guessed, is night blindness. That's kind of the first thing that happens where people just cannot recover even like basic sight at night. So that kind of shows you that your vitamin A stores are pretty low. And then the next sign is that these kids or people would get increased susceptibility to infections. And then in children, their growth is slowed just because, as we know, retinol is involved in growth. And then there's also gene expression, cell proliferation. So if you don't have enough vitamin A, basically you can end up to be a lot smaller in stature if you were greatly affected by this as a kid. And then the main clinical feature of this all has to do with the eye. So the main clinical feature is called xerophthalmia. And that basically just means an abnormality of the cornea. So things that you'll see are kind of drying and scarring of the eye. Usually looks like this. And then there's also billet spots that will occur. That's a white accumulation of slough cells on the eye. So if you end up working in countries where vitamin A does affect children, these are the main things the main way that they kind of just first diagnose a vitamin A deficiency. It's called xerophthalmia, so dryness, scarring of the eye, and then this white slough cell accumulation called a billet spot. In terms of toxicity, you'll notice that the TUL is set quite a bit lower than what we saw for the water-soluble vitamins. So you have a normal intake of 7 to 900. So it's at 3,000. So it's only maybe 4 times as much as what we're taking in before. And the name for the toxicity of vitamin A is called hypervitaminosis A. And different things occur. So if you do acute toxicity, so if you take in tons of supplements all at once, you'll get kind of typical effects. So nausea, vomiting, double vision, but your body will just try to get rid of it. So that's obviously where the vomiting comes from. Chronic effects, so people that take in kind of chronically high intakes of vitamin A, so 10 times the RDA, for several months, will start to experience hair loss, muscle pain, and liver damage. So one of the things that I always try to point out, and I think, as nutrition students, you probably already know this, but your clients may not. I think a lot of people see vitamins as just being these fun, healthy things, oh, vitamins are great, but they don't understand they're organic compounds and they can all be very dangerous. Remember, B6 was the one that could basically degrade your spinal cord. Vitamin A is very dangerous as well. But the main thing that vitamin A, where we see it kind of in the US, the toxicity symptoms are with birth defects. So teratogenic means birth defect. And this has been noticed when people are taking in about 4,500 micrograms per day, or RAEs, in pregnant women. So the main thing that happens is that babies are born without limbs if women are taking in very high amounts. But the scary thing is, if you remember, what was it, liver? If you're eating liver, I think it was 9,000 or something. So for women that are eating liver as a part of their food culture every day, it could actually be quite dangerous. And so, yeah, fetal malformations can occur. And this is, most people aren't getting this high amount from their diet. This actually occurred when the drug Accutane came on the market. And basically, Accutane is an acne drug that has really high doses of vitamin A, which I believe somehow vitamin A is helping to treat the acne. I didn't look up the actual mechanism, but they need the high doses of vitamin A in that medication because that's actually what's preventing the acne. Unfortunately, those high doses are also causing these birth defects if you take Accutane during the first trimester of pregnancy. So, from what I understand, and I know there's always a couple people in class that are taking these or have taken these types of medications. From my understanding, if you are prescribed Accutane, they make sure that you're taking some type of birth control the whole time. Like, I think in order to renew it, you have to show that you're on birth control. So that would help prevent these teratogenic effects. And then I'm often asked, so are carotenoids toxic? What happens with those? Well, remember, there's no DRIs for them, so there's no UL set. But they have noticed just when people take in kind of higher amounts, so like 180 milligrams per day, there's no serious health effects shown with like that level of intake. However, one thing that does happen if you're taking in, it's kind of, it's almost like cartoonish actually, but like there's something called hyperkeratinosis, which is when if you're taking in over 30 milligrams per day over the for a pretty long time, so for months, this can result in the yellowing of the hands and feet. So, literally, if you do eat too many carrots, you can actually turn your feet and hands orange or yellow. It's kind of crazy. I'm always surprised. Whenever I get to this slide, I'm like, oh, yeah, that is a thing. I've never seen it happen. I don't know if you have, but I'm sure this happens far more with people taking supplements because the average US person consumes about 1,000 to 4,000 micrograms per day of carotenoids, so 1 to 2 milligrams, or sorry, 1 to 4 milligrams, but you need 30 to start seeing the yellowing of your the fat pads on your hands and feet. This will, if you do happen to have a friend that's consuming this and this quantity and that happened, it's nice to know that that will go away when you stop consuming that amount of carotenoids. So it's not like a permanent thing. All right, let's do a quick mini review and then we're all done. So, what are the food sources of retinoids? So, the vitamin A form, retinoids. That one is animal sources. How about carotenoids? Plant sources. Yep. So remember, retinoids, animals, carotenoids are from plants. But also remember, carotenoids don't have a DRI and they're not essential. Function of retinol, OL. That's the one that's involved in growth. Function of retinal, AL. That's the night blindness one. And retinoic acid. That's the gene expression one. And then what is retinol palmitate or the retinol ester form used for? That one's the storage and transport one. Deficiency. So remember a bunch of eye stuff. It's called xerophthalmia. And toxicity. That was that hypervitaminosis A, the Accutane, and the birth defects, that type of thing. So high amounts can lead to birth defects. All right, well, I will see you at the next lecture. Have a great rest of your day.